Listen. We're good? All right. All right. Onwards and upwards. And quite literally in this case, right? We are literally moving to a larger scale. All right, we started at biomolecules, which are quite small, and now we're going to move on to cells, which you can see with your eyes sometimes. Okay, so bigger. Bigger is better. All right, so some quick reminders. And remember, I always try to pick up on things that we've talked about before. It gives us a nice footing to stand on. Organisms can be single-celled or multi-celled. So remember, we talked about one of the core things of being a living thing is having one or more cells, right? So you can be one cell and be alive, which means they're single-celled living organisms. We talked about these being largely bacteria or protists. Remember, protists are like those little swimmy guys that live in the water. Okay? And also don't forget that while we tend to think of bacteria being the bad guys, there are also non-virulent bacteria, okay? Living on your skin right now, living the best lives, you don't even know they're there. Okay, but of course, we've got some mammal bias. Okay, so multi-celled is what we are, and multi-celled tends to be what we think of. Okay, so animals, plants, and of course fungi, which we also tend to forget about unless we're eating it on a pizza. Okay. So there's a lot of variety. I'm actually going to use our mammal bias a lot in this particular unit because we are going to spend most of the time talking about our types of cells anyway. But we are going to look at both. All right. Roadmap, as promised and delivered. So we get a new roadmap finally, right? New road to be on. Okay. So we have a couple of different things that we're going to talk about. We're going to touch lightly on cell theory. Certainly, right? There is way too much of cell theory to discuss all of cell theory, so I'm just going to hit some highlights. And then we're going to talk a little bit about parts of cells in both of these things. Okay? We're going to highlight the things that we've talked about before. We've discussed cells in our biothemes unit. Right? We talked about cell theory then even. Okay? And we talked about cells in biomolecules. Right? We constructed a plasma membrane, which is important to cells. So we've got lots of platforms that we've already constructed for this unit. And so we're just really going to work on building the bridges between all of these platforms. So already you can see that as we go through this course, we really work on building up between units, right? And everything really is connected to each other. Okay. The bulk of this unit is really going to work on working our way through a cell. And we've spent a lot of time talking about some of this stuff already too. So we're going to start at DNA, okay? Work our way through making things like a protein, which we've already talked about as well. We're going to experience some new things, but ultimately end shipping ourselves out through the plasma membrane. So we're going to touch on, at least in part, a lot of familiar territory, which will be nice to continually get our feet on some solid ground as we continuously experience new things. All right, so let's start our journey. Looking at some cell theory, okay? So remember when we say what a theory is, okay, this is hypotheses and experiments and individuals and laws that have occurred over an extended period of time and have accumulated to create something that explains a broader idea, right? In this case, it happens to be, how do cells work and function? Okay, so this first started way back in the 1600s. So we have accumulated cell theory over about 400 years. And some stuff that's built into cell theory is even older than that. Okay, so one of the first guys that got on board is Robert Hooke, right? So we know that cell theory wasn't built by one guy. This guy's super dead. Right, so Robert Hooke, really amazing, right? Not technically a cell guy. Whoop. Okay, super into microscopy. Okay, with a level of patience that I certainly wouldn't know, looking at the kind of microscope he was using. Okay, so Robert Hooke, if you've not heard the story before, right, actually was just making thin slices of cork, right, because we just started to really understand how microscopes and lenses worked back then. And so when he made this thin slice of cork, you can kind of see it in the black circle up at the top there, he saw that when you slice into the cork, because cork's kind of bouncy and has air in it, that they had these holes in it, and the holes to him looked like prison cells. Okay, so he's got a dark sense of humor. But the cells, right, prison cells, so everything that had that shape was then called a cell. All right, so then as we began to identify organisms with the same shape, they were also called cells. And that then became enfolded into everything that was called cell theory later on. All right, so here's some more pieces that are key. Here's something that's familiar, right, as our first bullet, right? All living things are composed of one or more cells. Right, that was one of the very first things that we learned in class, right? You guys told me that, right? All living things have at least one cell in them. Right? And if you're a multicellular organism like you or I or your cat, right, then you just have more than one cell. Right, so there's some solid footing. Okay? All cells are formed from previously existing cells. Okay? We'll talk about this later in the semester. Excuse me. When we start looking at things like mitosis, right, the idea that one cell will divide and create two identical daughter cells, it seems intuitive to us now. Okay? Back in the days, well, we did not know, so we had all sorts of hypotheses tested. Perhaps cells are formed from crystallized water. You don't know until you test it, right? So we know now this is how it works, right? Your skin repels itself by, right, dividing the cells around it. So all your cells look the same, which we kind of like our arm to look like it's supposed to. Okay, this feel pretty good? Not so bad? No, that's, well, just know what I want. Okay. So now let's think about what dictates the size of a cell. Okay, we know for the most part cells don't get very big. Okay? Sometimes you can see them with the naked eye, but for the most part you have to look at them under some kind of a microscope, right? Particularly if you want to see anything good. Why is that? Okay. So let's start with how small cells can be. Cells can be pretty small, but they still can only be so small. Okay, and this one's more intuitive. So let's focus here for a hot second. So if we go back to the analogy that we have, where I'm carrying around a little plastic grocery bag, right? The plastic bag is my plasma membrane, and inside my plastic grocery bag is all my foodstuffs. And all my foodstuffs are all my important bits. Okay, DNA, organelles. Remember, organelles literally mean many organs. Okay, and we'll talk a bit later in this unit what exactly they do, but suffice it to say that many organs sound super important. Okay, so we got to keep them. Okay, I can only take that mini shopping bag and squeeze it so tight around my contents, right? can only come down so it's around my contents. I can't go any smaller than the stuff that's inside before I start squeezing all that important stuff out. And if it's really important, I can't squeeze it out, right? It's got to stay inside. So if I need to keep all this stuff, okay, my outside plasma membrane can only be as small as the stuff inside. Okay? Your skin can only be as small as your skeleton. Right? You can't go smaller than the stuff inside. It can get bigger, but it can't get smaller, right? You're limited by the stuff inside you. And so that's what we're saying here, right? You can't shrink your outer coat down smaller than whatever it's containing. Right? So we can't tighten that bag smaller than the groceries you're carrying inside it. That's not so bad. Kind of intuitive. Okay, I don't want to smush my breath. With me so far? Does this feel okay? Okay. The upper limit is a whole different beast. Okay, the upper limit uses what's called surface area to volume ratio. So we got to go back and use some math that nobody liked. All right, so let's get our terms under our belt first. Okay, so remember your surface area is the around. Okay, my volume is my inside. Okay? I like to think about my volume as the amount of fluid it would take for me to fill something. Okay, so I'm going to fill that cube. I want to think about if I stick a needle into that cube, how much fluid do I need to pump in there to fill that cube up? That's my volume. Okay, it's a nice visual. Okay, my surface area is just like how much fabric would I need to put around the outside. I was going to buy like a yard of fabric. Okay, those pretty easy visuals to work with. Kind of with me so far? Okay, here's why this matters. Okay, my volume, or that liquid that we're filling on the inside, is the alive part of the cell. Right? It's all the organs and the living bits. And so that part is like eating and surviving. So we need to feed the cell. Now, cells don't have things like mouths and booties like we do, right? So the only way you can feed a cell is by using the surface area, the fabric around the outside of the cell. With me so far? Okay. Which means that you need to have a decent relationship of fabric or surface area around the cell in order to feed, okay, without having too much filling, too much volume. Okay, if the volume gets too big, it'll starve to death. That intuitively makes sense without going too far? Huh. So here's our problem. If we take two examples, say a tennis ball and a basketball. Okay, the bigger you get, the bigger both your surface area and your volume gets. Okay, but your volume gets bigger faster. And I know that on the edge it doesn't seem that way, but let's picture it. Okay, so let's take my tennis ball. Okay. Okay, we're going to cover my tennis ball with fabric. That's easy to picture. It's kind of fuzzy anyway, right? I'm going to roll my tennis ball around in a puddle. Okay? And then I'm going to take all of that fluid. I'm going to fill the inside of my tennis ball with fluid. Okay, we want to picture how much of that tennis ball is actually going to fill with that fluid. Okay, maybe a third, maybe half-ish. Okay, so we're half hungry. Okay, let's do the same thing with my giant basketball. Okay, here's the beef. And we know basketballs are pretty empty, right? Because they make a bing when I hit the floor. Satisfying noise. That's why you watch basketball, for the bing, right? So if we cover my basketball in a thin layer of fabric and do the same thing, rolled it around in water, okay, and then I fill my basketball with the water from the outside, how full of fluid do you think inside that basketball would actually be? Wouldn't be very full, right? This is a big cavernous ball. And even though we're pretty big around, it's just super empty inside. Okay, so even though we've expanded around, the volume on the inside of my ball has just gotten too big. Okay, so we probably only filled like a quarter of this poor guy. That ball is starving to death. So this is what we're talking about here. We're talking about proportions. That as you get bigger, okay, that volume is proportionally getting larger, too large, right, for the surface area to keep feeding. Okay, and this is why you don't see 10-foot-tall cells walking down the hall going to school with you guys, right? Because they would starve to death. And this is also why you guys have specialized digestive systems to sort of make this work, so you can be, well, 6 feet tall and walking down the hall, living your best lives. I have an additional video on eLearn if this doesn't feel super comfortable, or to help you review. It has different surface areas of ice cubes to volume, and you can also see how terrible my husband is at holding a camera, that I highly recommend. We're also going to stop here because I know this particular thing can be a little overwhelming, so I don't want to add to it. So Fallon will have the socrative up. If you have any questions, also feel free to ask them. I'm not stopping that as well. I'm just letting you know I'm pausing for the day. Can you stop recording as well? Unless there are questions online.